Hello. Today's lesson is on DNA replication, and you should have received a blank piece of paper from either the sub or get it off the front counter. And go ahead, title your paper DNA replication. And I just drew two parallel lines and labeled them 5' to 3', and the opposite 3' to 5', because again, DNA is anti-parallel. I just drew one nucleotide, and you do not have to draw this nucleotide, but this was just to remind you where the 5' and 3' came from, that it was based upon the 5 carbon, the direction of the DNA oriented from the 5 carbon towards the 3 carbon. So again, you don't have to draw that, but just to remind you. Next, I'm going to draw the two parallel lines to show you that and how they represent a DNA strand. Now I'm showing that one line represents the sugar phosphate backbone. And if I were to put in these little rungs, like if it were a ladder turned sideways, that those lines would represent a base pair, such as adenine and thymine, guanine and cytosine. So if you want to draw that on your paper, and I think you should, go ahead, identify that the 5' to 3' line and the 3' to 5' line represent the sugar phosphate backbone. Okay, so I further drew out what these two lines represent. So hopefully you may recognize this better than just the two lines. So phosphate, sugar, phosphate, sugar, phosphate, sugar. It would continue, and then the bases coming off the sugar. So guanine, cytosine, attached to the sugar, adenine, thymine, both to the sugar, okay, and so forth. And then that would extend the whole way down. So now I hope you have an understanding of what just these two lines are representing. So now we will begin the start of DNA replication. Again, if it is clear to you what these two lines represent of the sugar phosphate backbone, you do not have to draw the additional, either say nucleotide or what was drawn here. So here's our DNA strand, and remember, a chromosome is just a long piece of DNA. So say this could be representing chromosome 5. So there's chromosome 5 before it is replicated. This piece is known as a centromere. But it's this strand of DNA is uncoiled. Chromosomes are coiled up DNA, but when DNA goes to replicate at a specific time during a cell cycle, it unravels. So it just becomes very long, strung out. And at certain places along the DNA strand is where it begins to unravel. It's called the origin of replication. I'm now going to draw these regions along the DNA strand. Okay, so along the DNA strand, there are areas that there are many base pairs of adenine and thymine. And this is where the DNA strand will begin to unravel. There's an enzyme that attaches along these sites of origin of replication, and this enzyme, helicase, latches on and starts to break the hydrogen bonds. And it's a lot easier, requires less energy to break at adenine and thymines because of only 2 hydrogen bonds between them, the base pair. So, and note how I wrote the ORI, origin of replication. They always write it as a cursive, and I'm not sure why. So we have all these sites along the DNA strand, and so that's where DNA replication begins, and it happens all along the strand at the exact same time. So this process actually goes very fast. All right, so hopefully you now have your sites of origin of replication added to your DNA strand. And now I'm going to add helicase, and I'll spell that out for the next strand.